Welcome, everyone. Thank you so much for joining, and I'm here to introduce our Arima Oncology Panel. Again, my name is Ibrahim, and I'm the Director of Product Management. During my portion of this talk, I'll be introducing Arima as a company and our 3D genomics technology platform. Then I'll go over how our 3D genomics platform is applied to cancer research, and then I'll follow up with the introduction of our new Arima Oncology Panel, which we are very excited about, and our early access program that we're initiating starting today. I'll conclude and wrap it up, and then hand it over to Dr. Atif Ahmed to talk about his exciting research in pediatric rhabdomyosarcoma. So just an introduction, Arima Genomics is the leader in 3D genomics. We've been around since 2015. And our what we have is our Arima technology platform. What we do is we use our core chemistry is centered around Hi-C, and this chemistry is delivered to our customers via kits and reagents or services and support. Ultimately, what we're trying to do is empower labs, molecular biology labs around the world to be able to bring 3D genomics into their lab. And so while we have kits and services, we really work with labs to make sure we find the solution that works for them. So why are we pursuing 3D genomics? So there's more to the genome than the sequence. Most legacy next-gen sequencing methods or genomics methods typically look at our sequence in a linear two-dimensional fashion. It treats our genome as if it's more like a computer program rather than the complex biological entity that it is. With our chemistry, we're capturing and retaining the three-dimensional information. And that information doesn't just provide the sequence, but the context of that sequence in proximity to other sequences. So you do definitely build that 3D structure of our chromosomes and our genome. And what that does is, it delivers multiple layers of information about our genome. At the highest level, it provides chromosome territories, which is very useful in developing digital karyotypes across of the genome. At the next level is chromatin compartments. So chromatin is organized into active A compartments and inactive B compartments. These compartments vary from cell to cell and can be useful in helping identify what is transcriptionally active and inactive. The next layer of information is topologically associated domains. These are regulatory domains that are conserved across cell types. A disruption of these domains can often lead to misregulation and is often seen actually in cancer. So this is a very useful level of information to be able to characterize. And at the most granular level are loops. And so within each TAD, DNA interacts with itself in various ways, either through histone binding factors or transcriptional factors. And this level of information is very useful in characterizing regulatory elements such as promoter enhancer loops. So these various 3D genome structural features, not just, as I mentioned before, have biological relevance. I already mentioned promoter enhancer communication, but it also has impact on coordinating transcription with splicing, coordinated gene expression, and DNA repair replication timing. In turn, we also have pathological implications associated with these features, and including long-range effects of single nucleotide variants, oncogenic fusion transcripts, and chromosomal translocations. Ultimately, Arima is trying to deliver 3 layers of information into one chemistry and platform. By virtue of it being a next-gen sequencing-based method, we are providing sequence-based information. But the 3D characteristics of our chemistry deliver structural information, which is useful in characterizing gene fusions and translocations, but also regulatory information, which is useful and to allow to detect 3D interactions to characterize insights with that impact the regulation of oncogenes. So why were we applying this? So just kind of talking about the application of 3D genomics in cancer and why we are pursuing this area. So cancer genomes contain 4 to 5 driver mutations in both coding and non-coding regions, and not all cancers have known drivers. What we do with our platform is we use 3D genomics to identify both known and unknown genetic and epigenetic drivers in cancer genomes. And so just to kind of go over a few examples of how our technology is able to do this. Well, this first example demonstrates how our platform is able to characterize structural variants to understand disease mechanisms. So in this case, I'm introducing a typical high-C heat map, which is a traditional way of visualizing high-C data. On this left-hand side is a normal karyotype, and you can see on the x-axis are cells, and in addition to the y-axis, and each cell represents a chromosome in our genome. And you can see along the diagonal that our chromosomes typically interact with itself, which is normal and expected in this digital karyotype. However, in an abnormal scenario where there is a structural variant, there is a high intense signal that comes across off the diagonal, which indicates interactions between different chromosomes. So because this is unexpected, this is evidence of a structural variant. The same heat map is visualized below to indicate how the chemistry is delivering this type of information, where we have our chimeric read fragments, which are linked together by virtue of our high-C chemistry based on their proximity. And so in an abnormal scenario, we're able to sensitively detect these structural variants by virtue of having multiple signatures of that variant spanning both sides of the variant. Typical next-gen sequencing or two-dimensional techniques require reads spanning across that breakpoint in order to characterize that structural variant. Our technology is able to do this specifically because we're not looking for one signature. We're looking for multiple signatures on both sides of that breakpoint. So a practical application of this is shown here in this case study. So we have a collaborator, Dr. Matiha Snudarel at NYU Langone, who had a pediatric patient with stage 2 glioma that was initially treated with a subtotal resection of the tumor. 6 months after the surgery, this patient experienced very rapid progression and the tumor returned quite aggressively. NYU performed comprehensive DNA and RNA sequencing of that primary tumor. And unfortunately, the result of that was inconclusive with no driver mutations identified. So there was very few options with regard to treatment. So Dr. Snudarel decided to apply our high-C chemistry on the sample just to see what additional information can be gleaned. And interestingly, analysis with the Arima technology identified a novel PDL1 structural variant in the sample, which every other assay had previously missed. This PDL1 structural variant, gene fusion, was confirmed with IHC and actually led to a change in treatment and an improved prognosis since there was a known treatment for this. And now, thankfully, the disease is now stable and the patient's his health has been improved. So this is a real-world clinical example of this technology being applied and something that Arima has been excited and proud of being able to be a part of. But in addition to characterizing structural variants, this is an example of how high-C can be used to characterize the regulatory profile of two different groups. In this case study, Dr. Ashinger Kaweka and colleagues compares the difference in 3D interactions between decitabine treated and untreated endocrine-resistant positive breast cancer samples. In the visual above, we are looking specifically at KRT8, where treatment with decitabine activates promoter enhancer communication when treated. And without treatment, there is minimal interactions. So the significance of this is that they were able to apply a therapy to activate tumor suppressor genes, which caused resistance to endocrine therapy. This approach can be extended by others to characterize regulatory differences between clinical samples types and unveil the mechanisms that impact treatment efficacy. And finally, this is an exciting paper that was released back in 2020 in Nature Genetics by Kloetgen and colleagues. What they were able to demonstrate in this paper was how the 3D organization of an oncogene relative to its enhancer could affect the progression of T-cell acute lymphoblastic leukemia. In this first set of diagrams representing the healthy patient, we can see that the oncogene MYC and this suspected enhancer element are 2 megabases away from each other. So due to that distance, it's difficult to characterize any sort of interaction between these two elements. And they're separated in linear space. And we can see over here in a high C profile, we can see that there is separation in a three-dimensional space as well. However, in patients with this specific type of leukemia that Kogan and colleagues are studying, there is an activation of the specific oncogene, and you can see based on 2D methods, it would be very difficult to associate this activation, this oncogene activation, with this particular transcription factor, specifically due to the distance between the two. However, with our 3D genome-based techniques, you can characterize this interactions based on their proximity, and you're able to kind of identify that there is this specific enhancer is actually activating this oncogene. This author of the studies went on to show that by inhibiting the transcription factor bound to this enhancer with a small particle pharmaceutical, they were able to restore normal expression of MYC. So using 3D genomics, they were, able to identify the mechanisms by which MYC is misregulated in T-cell acute lymphoblastic leukemia, and two, inform exactly which transcription factor needed to be targeted by the treatment to restore normal function. So ultimately, 3D genomics played a critical role in identifying the dynamic nature of oncogene regulation and informing therapy selection in human T-ALL. So all of these studies were built using the Arima technology platform, which consists of our core high-C genome-wide chemistry, but we also offer chemistry associated with high chip to do multi-omic chip plus high-C type studies. We also have a promoter capture, a panel-based approaches by virtue of having a promoter capture high-C panel and a custom capture high-C capabilities. We've also been involved in genome assembly studies with our collaborations with the Vertebrate Genomes Project and the Darwin Tree of Life project over in Europe. But we're very excited to share today a new offering in addition to this portfolio, which is the Arima Oncology Panel. So I mentioned in the previous slides our applications of our technology in this research and in cancer research. And so we're hoping to help drive cancer research forward with this panel by improving tumor classification and subtyping by virtue of being able to deliver breakpoint agnostic fusion and translocation detection, and the fact that it's FFPE compatible, so thereby enabling more discovery type research in a larger number of samples, particularly in rare disease states. Also, this panel aims to help identify novel drivers of disease by virtue of delivering promoter interactions with non-coding elements, and also by characterizing how non-coding variants can impact gene function by virtue of its interactions with the oncogenes on this panel. So, and then finally, the fact that it's a panel-based approach will help drive down the sequencing cost while maximizing discovery due to the size of the panel, which I'm going to go over in the next few slides. We can multiplex up to 4 samples per capture, and we generate rich data across all relevant cancer genes. So a little bit about the panel itself. It's precisely 1,404 genes for, across various different categories. So I wanted to point your attention to the categories visualized here. And just wanted to state that these numbers are not additive, but they are, because there's so many significant overlap between different categories. However, I wanted to call out that this panel includes 724 cancer targets that are found in the cancer gene census, and in addition to the classical Vogelstein hallmark genes, and that almost 700 of these targets are already associated with known treatments and therapies by being part of the actionable cancer targets list found in the MSKCC packed gene list, along with NYU and Stanford. So this is a true discovery-based assay to help improve tumor classification and to allow not just characterization of novel subtypes based on the regulation of the cancer genes in this list, but we also are able to help identify which of those genes could potentially have therapies potentially associated with them. So there are 2 design features associated with this panel. The first feature is the promoter targeting, and the next one is the gene targeting aspect of it. The promoter targeting feature of this panel has probes upstream and downstream of the transcription start site. And this enables the characterization of non-coding regulatory elements that might be impacting this particular gene. The gene targeting aspect has dense coverage over exons to have nice coverage over the coding regions of the gene, but also sparse coverage over introns. And this delivers breakpoint agnostic and sensitive fusion translocation detection. So in our initial studies, we are showing some data based on our control GM12878 samples. And we demonstrate that we can reproduce loop calls that correspond to functional genome characteristics, as in loops that are associated with active enhancer and H3K27 acetyl, which is associated with active promoter enhancer interactions. So you can see loops tied to those on both sides, and that the loop calls that we were able to generate in this data are highly reproducible. And the resolution we're able to achieve is 5 kilobase promoter loop interactions at just 200 million reads, which is significantly fewer reads than what is required for, let's say, a genome-wide based assay. Furthermore, we are able to, in this MCF7 FFPE control sample, we're characterizing a known gene fusion between ARID1B in chromosome 6 and TBL1XR1 and chromosome 3. And this heat map shows that translocation visualized here. And I think in the previous slide, I showed how a typical karyotype would have signal across a diagonal. So this is a zoomed in view of the chromosome 3 and chromosome 6 interaction, and how this signal indicates that there is indeed a translocation present in this portion of that chromosome based on this high C map. While we generated roughly 200 million reads on this sample, we downsampled to just 1 million reads, and we were able to see this translocation with just 1 million reads, despite having a panel that is 1,400 genes large. So we are very excited to have this panel being offered for early access now. Typically, we have a process internally where we have internal testing via alpha. We're active, which is now completed, and then the beta program is currently underway with our collaborators at Children's Mercy. Actually, Dr. Midhat Farooqi, who is on this call, is part of the beta program, and then we also have collaborators at St. Jude and NYU. And today we are activating our early access program, and the registration deadline is September 14th. There are only 20 slots available, so I would encourage you, if you are interested in learning more about our early access, to register now, and then we can reach out to you to kind of tell you more about the product and answer any questions you might have. The product launch is expected to be sometime in October, and so there is going to be some wait between those who receive the product first as part of the early access program and those who might have access to it later. So if you're interested, if you have your phone handy, you're more than welcome to scan this QR code or to visit arimagenomics.com/oncology-panel. So in summary, I wanted to wrap things up here. And Arima is a leader in the 3D genomics space by virtue of our optimization of high-C chemistry over the past 8 years. 3D genomics has led to progress in the characterization of disease mechanisms in cancer. We're very excited to bring the Arima Oncology Panel to the public now, which delivers rich 3D genome data across 1,400 cancer targets. And early access is now live, so sign up today and reserve your spot. I just also wanted to acknowledge the incredible team that has been working to develop this panel. They have been working for the past 8 months, closer to a year, to bring this chemistry and to kind of develop the gene list in the panel, so that way we can make this available to more researchers. So I just wanted to give a quick thank you to them.